is the largest of all the organelles. It's got the genetic blueprint for all cellular proteins. It responds to signals that dictate the kinds and amounts of proteins that are needed to be synthesized. Most cells only have one nucleus, they're uninucleate, but there are some, skeletal muscles in particular, that have many nuclei, they are multinucleated. And red blood cells do not have a nucleus at all, they are anucleate. The nucleus has three main structures: the envelope, the nucleoli, and the chromatin. There's an image of the nucleus with some rough endoplasmic reticulum present. The nuclear envelope is a double-layer barrier that enclosed a jelly-like nucleoplasm, the equivalent of cytoplasm. Outer layer, again, is continuous with a rough endoplasmic reticulum, and it has ribosomes on it. The inner layer, called the nuclear lamina, is a network of mesh proteins that maintain the nuclear shape and act as scaffolding for the DNA. There are nuclear pores that allow substances to pass in and out of the nucleus in a regulated fashion. Again, the nucleus is a selectively permeable membrane. Nucleoli are dark staining spherical bodies within the nucleus where ribosomal RNA is being actively put together. They associate with nuclear organizing regions that contain DNA that codes for ribosomal RNA, usually only one or two per cell, unless the cell is really involved in a lot of protein synthesis. The chromatin is about 30% of the DNA and 60% histone proteins, a little bit of RNA in there as well, being transcribed. It's arranged into fundamental units called nucleosomes, in which the DNA is wrapped around a histone. The chemical alterations of the histones can have an effect on expression of genes. We're learning more and more about that every day. And that chromatin will be condensed into chromosomes during the process of cell division. And that process of cell division brings us to the cell cycle, which is a series of changes the cell undergoes from the time it's formed until it reproduces. The life cycle can be divided into two major phases: interphase, which is carrying out its usual activities, and cell division, which we would call the mitotic phase, where the nucleus separates and the cell membrane separates in cytokinesis. So in interphase is the normal part of cell life cycle. If that cell is going to divide, then it will have subphases, which would include DNA replication. So all cells would be in G1, GAP1, and cells that cannot go beyond G1 are said to be in G0. The cardiac cells and nerve cells would be good examples of those. Then we'd have the S phase, which is the synthetic phase or the synthesis phase, where DNA replication, an entire copy of all the DNA, is made, followed by GAP2, in which we do the final preparations for cell